Before we study the classification of the viruses and different viruses one by one, we can just study the large DNA vaccines, large DNA genomes. There are very big viruses, as we have already studied, and there are again the very small viruses present on this earth. So there are different versatility of the viruses is present here. So we will study the large DNA genomes, what kind of the viruses have the very big large DNA genomes. Viral genome can be considerably big. For example, the double-stranded DNA genomes, and similarly they are genetically similar to the host cells, for example the adenoviruses and herpes viruses. Of course, it especially depends upon the type of the virus and type of the host. Genetically, the virus will be similar to its host. It's very important. If the host of the virus is big or the host of the virus has complicated genome, the virus will also have the complications inside its genome, and it will have the big or the double-stranded or single-stranded. Similarly, if the host of some typical virus is small or the genome is very simple, the virus will also be simple. Just like we can take the example of herpes virus, Herpes viridae family. It is very, very large family. It has about more than 100 members of its. 8 human viruses are very similar to overall structure. So herpes viruses have about near 8 viruses which are infecting to the human beings. So of course human being has very complicated genome. Similarly, the herpes virus will also have the complicated genome, and it is subdivided into 3 subfamilies. We will study in detail about every virus later. The subfamilies of the herpes viruses are alpha herpes viridae, and then beta herpes viridae and gamma herpes viridae. What is the alpha herpes viridae? Latent infections in sensory ganglion, and genome size is about 120 to 180 kb. Simplex virus, Virology, and human herpes viruses are the examples. Maybe human herpes viruses are also human herpes virus 1 and human herpes virus 2, human herpes virus 3 are the examples of this herpes virus. As we have already studied that herpes virus will have complicated genome because they are infecting the mammals and the human beings. Beta herpes viridae, these have the restricted host range. Their host range is very small as compared to the alpha herpes viridae. And genomic size will also be small as compared to 140 to 253. The genomic size is a little bit bigger than the alpha. Cytomegalovirus are the examples. Then muromelagovirus, rosimelagovirus, and similarly the human herpes virus 5, 6, and 7 are the examples which are contained in the beta herpes viridae family. Then the gamma herpes viridae, infection of the lymphoblastic cells, and its genomic size is much smaller as compared to the alpha and beta. It's about 105 to 175 kbp. kbp stands for the kilobase pair. It's the lymphovirus, and then the human herpes virus 8 are the examples of this gamma herpes viridae family. Then the herpes virus are the large linear double-stranded DNA genome containing about 35 viral polypeptides, and these polypeptides will of course encoding a large number of the enzymes, a large variety of the enzymes coding here. 2 covalently bound joint sections, unique long and unique small, and these unique long and unique small are measured together with the help of the inverted repeats. Allows the structural rearrangements of the unique patterns in this herpes virus. Then the herpes simplex virus is about 152 BP kilobase pair, and it's encoding about 80 genes are there. Its genome consists of 80 genes, densely packed with the overlapping reading frames. The open reading frames are overlapping because of its big size, and they are densely packed with the capsid. Each gene expressed by own promoter and extensively mapped by conventional genetic analysis. Most extensively studied virus until now is the herpes simplex virus. Then the large DNA genomes are also we have seen the adenoviruses. This is linear and again the double-stranded DNA genome, about 30 to 38 kilobase pairs of length and encoding about 30 to 40 genes inside. Terminal sequence is an inverted repeat of 100 to 140 base pairs, and denatured single strand from pen-handled structure. Pen-handled structure we will see later that what kind of the structures are these. And so here you can see that the size of the adenovirus is also very big, and it contains the double-stranded DNA virus. The pen-handle structures are important, are very important structures as far as the virus life cycle is concerned, and the replication, they are used in the replication. 55 kDa protein known as the terminal protein, and these are the pen-handle structures attached to 5 ends of each strand. In the genomic material, attached with the 5 ends of the genome genes, these are the pen-handle structures, act as the primer and initiate the synthesis of new strands. They are very important. At the time of the replication, they will be acting as primers, and that primers is important to initiate, as we will see in the practical portion, that how the DNA and RNA replication occurs in different viruses. So as a primer, the primer is very important and necessary to initiate the synthesis of new strands. As it's clear from the pictures, from the diagram given below, the linear structure and then the pen-handle structures are there, which are attacked at the 5 ends of the genes at that point, and they will just initiate of the replication. Expression will be done by two different parts. One is the promoters, which are present inside the virus, and then others are the splicing patterns.